Today I want to talk about something that's foundational to a lot of things you're going to learn later on. It's important to understand cell signaling, to understand aspects of physiology like the nervous system, the renal system, the endocrine system. So much of what you're going to learn depends on a couple of concepts from membranes and transport. So let's talk about it. Our membranes and the membranes of every cell are made of phospholipids. And phospholipids have a couple different parts. There's going to be a molecule connecting everything called glycerol. And label it with a G. And off this glycerol come two fatty acids. And that corresponds to this would be glycerol, and these would be the fatty acids. And you might recognize this from when we talked about the biomolecules, where you had triglycerides that had glycerol and 3 fatty acids. Here, in this one, we only have 2 because that third space is going to be occupied by a phosphate group. Now, if you're taking chemistry, which I hope you have, if you haven't, go and take it, you know that 8 means you have a negative charge on you. So, this molecule is what we call amphipathic, which means that part of it is going to be hydrophilic or water-loving. So that's going to be your phosphate. So this in the drawing that I drew, this here. And in this drawing, it's going to be these little gray things. And throughout the figures in the book that I'm using to construct these videos, it's going to be these little gray heads are the phosphates that are hydrophilic or water-loving. And then we will have our fatty acids and I guess our glycerol as well to some extent. They will be hydrophobic or water-fearing. So that's these, and in this drawing here. Those all do not like water. And the reason why that is because they are made of hydrocarbons. And carbon and hydrogen, they do not have any difference in electronegativity. That means that they don't have a dipole moment. One of them is not taking more electrons than the other one in their little game of tug-of-war with electrons. Whereas with water, you have oxygen who is totally hogging the electrons, which gives it a bit of a negative charge, and poor hydrogen is left without many electrons at all, which gives it a partial positive charge. And those partial positives and partial negatives don't really get along with things that do not have any form of charge, like hydrocarbons. So, this is why these phospholipids associate in a bilayer, or 2 layers, because all these hydrophobic tails, they want to be away from water as much as possible. So what's going to happen in this bilayer is all of the heads on either side, all the phosphate heads that are hydrophilic are going to face out toward the water, and all the hydrophobic tails are going to face inward and have their own little sanctuary away from big, or not so big, it's little, but scary water. So these membranes can vary in their fluidity, and that's due in part to the saturation of these fatty acids. Now, what is saturation? Saturation has to do with how many double bonds you have. So I'm going to go into a little bit of chemistry. If you have, let's say, little ethane, this is going to make sense in a minute. You've got 2 carbons and 6 hydrogens. And these carbons make 4 bonds, like carbons always make 4 bonds. What happens if I add a double bond? Well, these carbons say I can't have 5 bonds. And so things that have a double bond, you have to take away some hydrogens. So all of a sudden, this is going to have less hydrogens. So when we say saturated, we mean saturated with hydrogens. That would have been the single bond, where we have as many hydrogens as we can fit on there. With double bonds, we call them unsaturated, because you do not have as many hydrogens as you could if you had a single bond. So, going back to our phospholipids, if this is glycerol, if you have a fatty acid that has only single bonds, it's pretty much going to go straight. It's going to be kind of boring. But if you have a double bond, things are going to get interesting, because double bonds change the geometry of this molecule. So instead of going like this, it's going to go like this. And that's going to affect the membrane fluidity because all of a sudden we've got a little bit of space here, right? So if you take a look at this diagram right here, this membrane is more fluid because it's got more unsaturated lipids or unsaturated fatty acids. If you have only saturated or only like this kind here, then you're going to have something that looks like this. It's going to be a lot less fluid. It's going to be rigid, or they call it viscous in this figure. The classic examples of saturated and unsaturated fats that might be more recognizable to you are things like oil is an unsaturated fat. It is more fluid than, say, butter, which is made with saturated fat. There's another component to membrane fluidity, and that is cholesterol. Cholesterol is a steroid, which means that it's got this structure with 4 fused rings. And it's more complicated than that. It's got some extra things on these rings that I haven't memorized, so you don't have to memorize. But the point of it is that it kind of sits in some cell membranes. And when things get too warm, or there are too many unsaturated fats, like the membrane is getting too fluid, cholesterol regulates this by limiting the movement of these fatty acids. Whereas when it's really cold, or when you have lots of unsaturated fats, this cholesterol is going to create spaces in the membrane and make sure there is room for movement. So cholesterol is going to regulate membrane fluidity and keep it in this happy medium. So our cell membrane is more than just these phospholipids. People have called our cell membranes a fluid mosaic. And this means that, the mosaic part, it means that stuck in this membrane all over are proteins. And some of them hang out on the outside and some of them go straight through. And the fluid part means that things don't just stay where they are. They're going to move around laterally within the membrane. So here's kind of a model of what the cell membrane might look like if you were to shrink yourself down to the size of a phospholipid. So we have our lipids. We have some proteins. All the proteins in this book are going to be purple. These figures that I'm using. And we've got some sugars attached to them. And this whole bit out here is called the extracellular matrix, or the ECM. And it's just a network of proteins and sugars that is going to kind of communicate with the cell's outer environment and connect the outer environment to the membrane proteins, which are going to connect everything else to the intracellular environment. So we've got some names for these guys. These proteins are called integral proteins because they are integrated within the membrane. And then these ones we're going to call peripheral proteins because they're not in the middle of the membrane, they're on the periphery. Now, we talked about fluid mosaic. Fluid mosaic, the fluid part means that things are going to move around within the membrane. And there are some guys who wanted to figure out how this worked. And they did this experiment where they took 2 cells and they fused them together and they marked them with different proteins before fusing them together. So if we hypothesized that this membrane, the proteins stay exactly where they are and they don't move, they are anchored. What would happen when these 2 cells get together? Well, the new cell we would expect to have pink. I don't have pink, I have red, so I'm going to use red. These red or pink proteins on this side of the cell membrane and the purple ones over here, and they wouldn't be intermingling because things don't move around in our hypothesis. Well, our hypothesis turns out to be wrong, and they found that these proteins are going to kind of mix with each other. And that's because things can move around within the membrane. And that's the basis of the fluid mosaic model. So now that we have a bit of an idea of what the membrane looks like, let's figure out how things get from one side of it to another and how the cell regulates what goes through. So really foundational to learning about membrane transport is this concept of diffusion. If you've taken chemistry before, you might have heard of diffusion. It's the movement of molecules, the net movement of molecules from high concentration to low concentration. And it's not like this atom is going to sit here and go, I am telepathic. I can sense that on the other side of this solution or this medium or whatever it is, there are less of me, so I'm going to go there. Everybody, all these atoms or all these molecules are going to be moving around randomly. And they're going to be bumping into the walls or the membranes of whatever container or container-like thing that they're in. And they are going to be moving in whatever random direction the solvent takes them. This is going to result in them spreading out. Like, imagine if you put a cube of sugar in water, it's not going to stay a cube of sugar. The sugar is going to dissolve and diffuse out across the whole entire thing. It's not like it dissolves and then just stays in one spot, and when you drink your tea, it's all bitter and then one sip is sweet. Although gravity does do that sometimes. But the point is that things are moving in all directions. And eventually, this means that you will reach an equilibrium where the movement of things to one side, so like the movement of things to the left in this diagram, is going to be pretty much equal in rate to the movement of things to the right side. So that's the concept of diffusion. And there's a few different ways that the membrane of your cells allows this to happen. One way, we call it simple diffusion. And simple diffusion means that things can go right through the membrane on their own without any help from any type of membrane protein. And remember that the middle of this membrane is hydrophobic. It's full of hydrocarbons that do not like being around charges or partial charges. So the things that are going to be able to do simple diffusion are very, very limited. Number one, you have to be small, because the gaps between these lipids are not very big. Number two, you have to be nonpolar. So the main two molecules that are going to go across the membrane like this are O2 and CO2. Because if you look at these molecules, they're exactly symmetrical. Electrons are going to be getting pulled to one side as much as the other. You're going to end up with no net dipole moments. Steroid hormones can also come through here. So if you have something that is like a hormone and it's hydrophobic, like we talked about the 4' fused rings, things that derive from cholesterol, they're going to be able to get through. So if you remember that cholesterol is a part of the cell membrane, then it might be easy to remember that things that are derived from cholesterol can get through if they want to. Anything that's larger or anything that's charged or partially charged is going to need to use a protein to get through. And this includes water. So water or any polar or larger solute is going to use either a channel or a carrier, and it's going to either require energy or not. When you don't require energy, we call this passive transport. It's also sometimes called facilitated diffusion. Now, in the different classes I've been in and taught, teachers will switch up the language that they use. Some classes will say facilitated diffusion is going through a channel protein. So like this, where the protein just has a hole in the middle and things float through the hole in the middle from high concentration to low concentration. So some people will call that facilitated diffusion, and then passive transport will be this carrier protein that kind of, this one doesn't, but a lot of the ones I've seen look like a Pac-Man, and they'll bind specific things, and then they'll have what's called conformational change, and conformation just means shape. So they're going to change their shape and let things out on the other side of the membrane. So this would be the membrane. So my point is, sometimes I've heard the first thing, the channel, being called facilitated diffusion and this being called passive transport with the carrier. Sometimes the names are interchangeable, sometimes it's the other way around even. So I would just say pay attention to your class and to what your professor or your teacher is saying. If you're taking a standardized test such as the MCAT, in all likelihood, you're not going to be tested between these two types of transport. It's going to be more the category of passive transport and the category of active transport. So what is active transport then? Active transport is going to be the transport of solutes against their concentration gradient. So from low concentration to high concentration. And since this is not something that happens naturally or not something that happens spontaneously, we are going to need some energy in order to do this. In cells, the primary energy source that can be used conveniently that we make out of our fuel is ATP, adenosine triphosphate, and this gets used by turning it into ADP. So we are releasing a phosphate via hydrolysis. So we have water that goes into this and does not come out. And this is going to help us get things across the membrane the opposite way from where they would, quote unquote, where they would want to go spontaneously. One of the famous examples of active transport using ATP is called the sodium-potassium pump, and this is essential to maintaining the electrical potential in a resting neuron or nerve cell. So this one is going to pump both sodium and potassium in one go. It's going to pump 3 sodiums out of the cell and 2 potassiums in. You can remember this in a couple of different ways. Because sodium comes first in the name, that can be your cue that there are 3 sodiums, there are more of them than potassiums that are 2. The other cute little acronym that I love is pump K in. It spells pumpkin, but that's how you know that potassium, or K, goes into the cell and sodium goes out of it. Anyways, if you're just starting to learn about membranes, that's probably not important right now to you, but if you're going to learn about neurons or physiology or anything a little more advanced, it's going to come back and bite you, so make sure you know about it. Another type of active transport is coupling or cotransport, and in cotransport, we have one thing that's going with its concentration gradient, or from high concentration to low concentration, and another thing that's going against its concentration, from low concentration to high concentration. So the one that's going with its concentration gradient, it's losing energy because it's going from a high energy state of it's all on one side to a low energy state of it's all spread out. We're going to use this energy in order to get the other molecule to go against its concentration gradient. So in this example here, hydrogen ion or proton is going with its concentration gradient. There were lots of them out here, and maybe 1 or 2 of them in here, and this is going this way, and it's releasing energy. So not ATP, but some form of chemical energy, and that's going to be used to drive this gradient of taking a sugar the other way, to go against its concentration gradient to where there's more of them. So that's membranes and transport. Oh, wait, there's one more diagram, because we've gone over how little things get across, but how do big things get across? And this is also essential to a lot of stuff you're going to learn later on. So sometimes cells need to take big gulps of what's outside of them, and they can do this in a few different ways. There is phagocytosis. Phagocytosis means eating of the cell. Phago, eat, cyte, cell. And this is going to be used often in the immune system. You might have heard of macrophages. Macrophages and other phagocytic cells are going to eat pathogens like bacteria and then spit out little parts of them for your immune system to recognize. Maybe some cells, like some unicellular or smaller eukaryotes, this is their primary way of getting nutrients. So basically what's going to happen is the membrane is going to project these little pieces of it using the cytoskeleton to support it. It's going to project these pieces of the membrane outward, and it's going to literally engulf this thing. So it's going to reach membrane around until the thing is covered in membrane, in a little sac of membrane, and we call this little sac of membrane a vesicle. There's another kind called pinocytosis, and pinocytosis means drinking of the cell. Pino is drinking, and cyte again is cell. So in this one, we're just going to be taking smaller gulps. Maybe it's not like a giant particle like a bacterium or like a big granule of food. Maybe it's just the solution around the cell when the cell senses that it needs more solutes. A third thing is called receptor-mediated endocytosis, and this one means we want to uptake a specific molecule. So, for example, with the receptor-mediated endocytosis, when your nerve cells or your neurons connect to each other, they connect at something called a synapse, which is a gap between the two cells. So this cell that's been sending a message down the length of the cell is going to send out things called neurotransmitters. And this is what lets the other cell know by binding to receptors on this end that it needs to send a message on to the next neuron after it. But how do we keep this from going on forever? Because if, let's say, this is in my hand and I touched a burning stove, and now my neurons want to let me know that my hand is burning, I don't want to think that my hand is burning after I've taken it off. So how is my body going to get rid of these neurotransmitters going through this little gap, which is known as a synapse? Well, it's going to use receptor-mediated endocytosis, so it's going to make an infolding of the membrane, and it's going to have receptors that are specifically shaped to bind this neurotransmitter, the molecule that was signaling between these two cells, and it's going to bring it back into the cell so that the synapse or the gap between these two cells is not filled with these neurotransmitters anymore. So it just works by things binding together using any number of intermolecular forces that we discussed when we discussed proteins and quaternary and tertiary structure. Those same forces are going to be used in this. And it's going to make a little vesicle and pull these molecules back into the cell. Now, this process of endocytosis, which is kind of collectively what we call these things, where you bring something or a bit of solution into the cell by pinching inward, it's got an opposite. The opposite is called exocytosis. So this was endocytosis. And this is going to be exocytosis, otherwise known as secretion. So the main process is happening right here. You've got a vesicle inside, and it's going to fuse with the membrane. If this thing will ever let me write. There. It's going to fuse with the membrane, and it's going to let out its contents on the outside of the cell. If you are learning more higher level biology, you're going to want to learn about this whole path where things can start out being made by bound ribosomes in the endoplasmic reticulum. They're going to secrete these chemicals, these proteins specifically, into the lumen of the ER. And then vesicles can butt off from the ER, fuse again with something like the Golgi apparatus. The vesicles can butt off of the other end, fuse with the membrane, or any other organelle, but here our focus is exocytosis, so a vesicle can fuse with the membrane and secrete whatever is inside of it. And this is really helpful for, say, if we want to send a hormone. If I haven't eaten in 7 hours because I've been teaching way too much, then what's going to happen is there are going to be cells in my pancreas that are going to secrete a hormone called glucagon. And the way that they do that is they're going to have it in a little vesicle, and it's going to fuse with the cell membrane, and out comes the glucagon into my bloodstream, and it can tell my whole body, you need sugar. And that's how endocytosis and exocytosis work. So I hope this video is informative. Now you have a better idea of membranes and transport.